So let's talk about the structure and function of DNA. So DNA stands for deoxyribonucleic acid, and let's examine the structure and the function. So DNA is the genetic material which is transferred from parents to children and from generation to generation. DNA is a chain of nucleotide which is joined together, and a nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. So the sugar, as we said, is deoxyribose, and there is a phosphate group and there is a nitrogenous base. The nitrogenous bases are adenine, guanine, cytosine, and thymine. Adenine and guanine are called purines, and cytosine and thymine are called pyrimidines. There's another nitrogenous base, but that's associated with the RNA, which we'll talk in another lecture. And in this slide, you can see the DNA structure, the chemical structure of the DNA. And you can see there is a phosphate group, there is a sugar group, and there is a nitrogenous base. And here it is adenine. And on the right side of the slide, you can see the other two nitrogenous bases, which is adenine and guanine, which are purines. And below that, you can see cytosine and thymine, which are the pyrimidines in DNA. And finally, you see uracil, which is also a pyrimidine, but that's in the RNA. So the structure of DNA was discovered in 1953, but as early as 1900, the components of DNA were known. So we knew that it was made of a sugar, phosphate, and a nitrogenous base, but it was only in 1953 that the structure was discovered. This was discovered by Watson and Crick, who proved that DNA had a double helix shape. Double helix means that it resembles a staircase, which you can see on the right side. This is the structure of the DNA. So the structure of DNA shows that it has a sugar phosphate backbone on either side, and it's connected by steps of the staircase. The steps of the staircase are pairs of nitrogenous bases, and the nitrogenous bases are united by hydrogen bonds. So here you can see the hydrogen bonds connecting the nitrogenous bases. So in the nitrogenous base, you can see there is a base pairing, and the purine on one strand is bound to the pyrimidine on the other strand. So here you can see the adenine is bound to the thymine, and it's connected by two hydrogen bonds. In the second step, you can see the guanine is bound to cytosine, and there are three hydrogen bonds. And again, the next one is thymine, which is attached to the adenine, and the next one is a guanine attached to the cytosine. So you will notice that a adenine always binds with a thymine, and a guanine always binds with a cytosine. This is called the law of complementary base pairing. So if you look at the function of DNA, you see the main function of DNA is to carry instruction for synthesis of protein. For this, there is a process. From DNA, a messenger RNA is generated, and it is the messenger RNA that creates the protein. So what is a gene? A gene is a region in a DNA that has a code for synthesis of a specific protein. So not all regions in the DNA have a code for protein synthesis. So it is only the gene which has that particular code. So gene is the protein coding segment of the DNA. So humans have about 20,000 genes, or they have about 20,000 protein coding segments. This 20,000 actually represents only 2% of the total DNA. The remaining 98% is non-coding. But they have some other important roles to play. So the 98%, which is non-coding, plays a role in chromosome structure and also regulates the gene activity. So if you look at the chromosomes, some chromosomes are gene rich. That means they have a lot of codes. They code a lot of proteins. And some chromosomes are gene poor. Gene poor chromosomes code for only a few proteins. Humans have 23 pairs of chromosomes. That's a total 46 chromosomes. You have 23 pairs of chromosomes because you inherit one set of chromosome from mom and another set of chromosome from dad. And that is why you have 23 pairs. The 23rd pair is called a sex chromosome. That determines the identity of the individual. In a non-dividing cell, the DNA exists as chromatin. Chromatin are fine filamentous material, and the chromatin is wrapped around histone proteins. The long threads of chromatin are packed into the nucleus, and the histone proteins are crucial for packing chromatin into the nucleus of the cell. So that's all about the DNA. So we discussed the structure of DNA, and we also discussed the function of DNA. Thank you very much.